Robert Hooke was an English natural philosopher who played an important role in the scientific revolution. He was called the father of microscopy. Hooke discovered plant cells in 1665. What he saw, like in this picture, were the cell walls in cork tissue. He was the one who named them cells. The box-like cells of the cork reminded him of the cells in the monastery. Another scientist named Antony van Leeuwenhoek, commonly known as the father of microbiology, have made over 500 microscopes and created over 400 different types of microscopes, but only 9 still exist today. Theodor Schwann, Matthias Schleiden, and Rudolf Virchow proposed a cell theory. All living organisms are composed of cells. The cell is the basic unit of life. Cells arise from pre-existing cells. There are 2 types of cells, prokaryotes and eukaryotes. Let us start with the prokaryotic cell first. One example is the bacteria. Prokaryotes are organisms that don't have a cell nucleus or other membrane-bound organelles. Most are unicellular, which means they only have one cell. Eukaryotes are organisms whose cells contain complex structures enclosed within membranes. They are typically much larger than prokaryotes. The picture that you can see here is a structure of an animal cell. They have a variety of internal membranes, organelles, and cytoskeleton. Cytoskeleton is composed of microtubules, microfilaments, and intermediate fibers. It is a web of protein fibers. Another organelle in the eukaryotic cell is the nucleus. It is surrounded by a double membrane called the nuclear envelope. It contains DNA, which the instructions are stored in there. The Golgi apparatus, or the Golgi complex, is a set of flattened, membrane-bound sacs. The function of it is to modify, sort, and package cell products for distribution. The lysosome is a vesicle that contains specific enzymes that break down large molecules, and they help recycle materials. A vacuole is a fluid-filled vesicle found in the cytoplasm of plant cells. The endoplasmic reticulum, or called the ER, is a system of internal membranes that moves proteins and other substances through the cell. There are 2 kinds, the rough ER and the smooth ER. The rough ER has ribosomes attached to it, and their function is to make proteins. The smooth ER doesn't have ribosomes, and they make lipids and break down toxic substances. By controlling the movement of substances that move in and out of the cell is one way that the cell maintains homeostasis. The cell membrane is made of a sea of phospholipids. A phospholipid is a specialized lipid made of phosphate head and 2 fatty acid tails. The head is polar and is attracted to water, or hydrophilic, while the tails are nonpolar and are hydrophobic. As you can see, the phospholipids form a double layer called the lipid bilayer. Just read the following slides to know more about membrane proteins. Cells communicate and coordinate activity by sending chemical signals that carry information to other cells. The meaning of signal is anything that serves to direct, guide, or warn. Just read the following slides. Organisms use and store energy in chemical bonds of organic compounds. Almost all of the energy in organic compounds comes from the sun. Autotrophs are organisms that are able to perform photosynthesis. They make organic compounds that serve as food for them and for almost all of the other organisms on Earth. Most autotrophs have a supply of food as long as sunlight is available. The picture here is a diagram of a basic photosynthesis, and we learned this in grade 6. But as you go to grade 9, it gets more complicated. There are actually two types. Light reaction and dark reaction are called the Calvin cycle. Let us start with the light reaction first. Inside the chloroplast, the space inside the inner membrane is the stroma. Within the stroma is a membrane called the thylakoid membrane. Water splits first to form two hydrogen ions and an oxygen molecule to the concentration gradient. The electrons from the water goes to photosystem 2. Electrons get excited by two photons of light. Plastic quinone QB carries the electrons plus two protons to another electron transport protein. Protons are released, and a hydrogen ion is pumped into the thylakoid using energy from the electrons. The electrons are transported to the photosystem 1 and are re-energized by light. NADP plus hydrogen ion and electrons form NADPH. A hydrogen ion is diffused to the ADP synthase, and that energy is used to combine ADP or adenine phosphate plus phosphate to make ATP or adenine triphosphate.